Hello everyone. Today we'll discuss about the classification of the enzymes, coenzyme and metalloenzyme. Now first of all, the classification of the enzyme is mainly done on basis of 6 classes of the enzymes. Now out of which first one is the oxidoreductases enzyme. Now as the name says oxidoreductases, that means it will do oxidation and the reduction of particular substrate and the coenzyme simultaneously. That means catalyze oxidation of one substrate with the simultaneous reduction of the other, or we can say the coenzyme. Over here, as you can see in this example, the ethanol or the alcohol in the presence of this ADH named enzyme known as the alcohol dehydrogenase enzyme will undergo oxidation and will form acetaldehyde. Right, over here the ethanol is undergoing dehydrogenation or the oxidation, while the NAD coenzyme is undergoing this reduction process and forming NADH. Okay, the presence of ADH. Right, now other examples of this are the lactate dehydrogenase, G6PD means glucose 6-phosphate dehydrogenase, succinate dehydrogenase and dioxygenase. Next, class 2 is the transferases. Now over here the enzyme actually transfers any one group other than the hydrogen from one substrate to another. Previously we were transferring the hydrogen, right, from ethanol to NAD, but over here this transferase will also transfer the particular group only other than hydrogen. Now for example, as you can see over here, this is the hexokinase enzyme. Okay, and this hexokinase enzyme, what it will do, as you can see, this is the glucose, right, or hexose. Now this hexokinase will take the ATP and will transfer the phosphate group, one phosphate group of ATP in the presence of magnesium, to the glucose. So it is transferring one particular group to the other substrate, right, and will form this glucose 6-phosphate. Other example are the amino transfers. So just remember, transferase will transfer one group other than hydrogen from one substrate to another, as seen over here from the ATP to the glucose. Now the hydrolysis, as the name says, hydro and lase means breakdown of the particular compound, okay, with the help of water. So they hydrolyze ester, ether, peptide or the glycosidic bond by adding water. So in class 3 hydrolysis, as you can see over here, acetylcholine esterase. It will actually hydrolyze this acetylcholine. Okay, it is a neurotransmitter. So acetylcholine, it will hydrolyze it with water, okay, and will form acetate and choline. Now this example I have discussed about this in my nerve video of the physiology. This cholinesterase enzyme is mainly present in the palisades of the particular postsynaptic membrane in the neuromuscular junction. At the postsynaptic membrane, the palisades are present, and in that palisades, this acetylcholine esterase enzyme is present. We actually cleave this acetylcholine into acetate and choline. Now, other example are trypsin in the GIT. Now, the lyase, that means it will cleave something, right? But they remove groups from substrate or break the bonds by mechanism other than hydrolysis. Means without adding water, they will cleave the particular bond. For example, as you can see over here, fructose 1,6 biphosphate. In the presence of aldolase, it will cleave this fructose 1,6 biphosphate in the absence of water and will form DHAP, dihydroxyacetone phosphate, and GAP, that means glyceraldehyde 3-phosphate. So dihydroacetone phosphate and glyceraldehyde 3-phosphate actually formed by the fructose 1,6 biphosphate. Other example over here are the HMG-CoA lyase and the ATP citrate lyase. Now fifth is the isomerases. As the name says, isomers, so they will produce optical, geometrical or positional isomers of the particular substrate. Okay, and the examples are racemases, epimerases, cis transfer isomerases, and over here as you can see, DHAP, dihydroxyacetone phosphate, will be converted or will be isomerized, okay, to GAP, D-glyceraldehyde 3-phosphate in the presence of triose phosphate isomerase. So this will mainly particularly form the isomer of particular substrate. Now last 6 is the ligase. Ligase means they will link two particular substrate. They link two substrate together usually with simultaneous hydrolysis of ATP. So acetyl-CoA plus CO2. Okay. In the presence of acetyl-CoA carboxylase will form malonyl-CoA ADP plus PI. Okay. In the presence of 1 ATP, we will hydrolyze 1 ATP over here, and with that particular energy, the acetyl-CoA is converted into malonyl-CoA. Other examples are the glutamine synthesis and the PRPP synthesis. So just remember, in the ligase, the acetyl-CoA and CO2, these two are actually ligated by the particular acetyl-CoA carboxylase enzyme. So these were the 6 classes of the classification of the particular enzymes. Now what are coenzymes? You may have heard about this. Now you know that the enzymes are maybe simple protein or complex protein. In the simple protein, they are having particularly amino acid groups and the protein structure, and they are known as the apoenzymes. The simple protein having no non-protein structures are known as, or no complex structure are known as the apoenzyme. While what is prosthetic group? If a particular enzyme is having a non-protein compound in it as well, it is known as the prosthetic group and it is known as the coenzyme. And remember, apoenzyme plus coenzyme forms the holoenzyme. Okay. Now coenzymes, this coenzyme is further divided into two groups. First of all, those group that take part in reaction catalyzed by the oxidoreductases by donating or accepting hydrogen. Remember, over here there will be oxidoreductases enzyme, means there will be taking or the giving off of the hydrogen. While over here, those in the second group, those taking part in the reaction transferring group other than the hydrogen. Now in the first of all, group 1 of coenzyme, that is the oxidoreductases, or the group having reduction and the oxidation process by taking or giving up the hydrogen. Now what happens in this, the changes occurring in the substrate is counterbalanced by the coenzyme. As you can see over, for a very easy example is this one, lactate. In this, as you can see, the lactate is actually being oxidized, or the hydrogen in that it is actually released by the lactate dehydrogenase or the LDH enzyme. LDH enzyme will remove the hydrogen from lactate, okay, and will cause the oxidation of this particular lactate. But simultaneously at that time, this NAD coenzyme is actually reduced, and this NAD will take the hydrogen of this particular lactate, and then the lactate will convert into pyruvate. So in simple words, what happens is, whatever reaction is done by the substrate is actually counterbalanced by the particular coenzyme, and that's why in the first group of coenzyme, this particular coenzyme is also known as a co-substrate, because it is counterbalancing the reaction, and also known as a secondary substrate. Now other examples are NADP, NADPH, FAD, FADH2, FMN, FMNH2. Now nicotinamide adenine dinucleotide NAD. What is this? It is a coenzyme we studied over here. Now we study about this in particularly in detail. Now this is actually synthesized by the nicotinamide, and remember, it is a member of vitamin B complex, and the structure of it is nicotinamide, ribose, 2 phosphate bonds, ribose and adenine. Now over here, as you can see in the previous example only, the lactate is oxidized, simultaneously NAD is actually reduced to NADH. Now what happens over here is, this lactate in the place of LDH will 2 hydrogens are released, right? Now out of this 2 hydrogen, of course, whenever we will cleave this hydrogen, we will get 2 H plus and 2 electrons. Now out of this 2 electrons and 1 H plus will be taken up by the NAD plus, and it will form NADH. Well, the 1 H plus is actually released in the surrounding medium. Okay. Now next group is the second group of coenzyme. That means the group that actually transfers group as apart from the hydrogen, right? Now these coenzymes take part in reaction transferring group other than hydrogen. Now remember, a particular group or the radical is transferred from the substrate to another substrate, and most of them belong to vitamin B complex only. Now the example of the second group of coenzymes is the ATP adenosine triphosphate. Now we all know ATP is considered to be the energy currency in the body, and in the ATP molecule, the second and the third phosphate bonds are high energy bonds, and during the oxidation the foodstuffs, energy is released, and a part of which is stored as chemical energy in the form of ATP. The endergonic reaction are carried out with the help of energy released from the hydrolysis of the ATP. So whenever we will do the hydrolysis of the ATP, the energy release is mainly used for the endergonic processes. Now as you can see in this example, glucose. The glucose is actually converted to glucose 6-phosphate by the enzyme hexokinase in the presence of ATP, where one molecule of where one phosphate compound is transferred to the glucose. So these are some of the examples of the coenzymes and the groups transferred by them. Now the metalloenzyme, as the name says, the enzyme will require metals for their activity. So over here, so some of the example of these are, so these are some of the example of the metals and the enzyme containing the metals. The metal tightly bound to the enzymes are the copper with the tyrosinase. Okay, copper and tyrosinase actually get tightly bound to the enzyme. Now over here, in some cases, what happens is the particular reaction is actually going smoothly and the enzymes are working well. Okay, but when the ions are actually added or the metal ions are added, it will enhance, it will actually increase the activity of the particular enzyme, and these particular enzymes are known as the ion activated enzyme. For example, calcium ion will activate the pancreatic lipase. Now some of the non-metal ions can also be act over here, and example are the chloride ion activate the amylase. A metal ion participate in reaction in mainly three ways: by binding to the substrate and orienting them for reaction, simply binding and orienting the reaction; by mediating redox reaction through the reversible changes in the metal oxidation state; and by stabilizing the electrical charges. As the cofactors, the term is collectively used to include the coenzymes and the metal ions. And remember, the coenzyme is an organic cofactor. So this was all about the classification, Metalloenzyme and the cofactor and the coenzyme. We will discuss about the particular isoenzymes and the factors influencing the particular enzyme activity in a further next videos. Hope you did well. Thank you so much.